One of the most important questions in biology is how an organism is able to reproduce, and humans count as organisms. So let's say we've got a couple of people here. Let's say this is a man with a nice bushy beard, and this is a woman who has shoulder-length hair. Well, what needs to happen? These parents need to be able to pass on their genetic material to the next generation. What does that really look like? What is genetic information to a human? Humans carry almost all of our genetic information in the chromosome. So what's a chromosome? A chromosome is a mix of DNA and protein. So it's one double-stranded piece of DNA, really long, but actually a single molecule, which is very long, and some proteins that get associated with it that help the DNA function and fit into spaces in a reasonable way. So here's a representation of what these parents' chromosomes might look like. You'll notice that these are all paired up. These are called homologous pairs of chromosomes. So again, the question is, how do these parents take all of this genetic information, all this genetic material, and give it to their kid? So first guess is that these parents are just giving the kid all of their DNA, and the kid ended up walking around with twice as much DNA as the parents. You can probably see right off the bat that this would be kind of a problem because every generation would have twice as much DNA as the generation before it. Maybe each generation would be able to get rid of some of the DNA to keep things sort of stable. But anyway, that's not what happens. The parents don't give all of their genetic information to their offspring. They only give half of it. So each parent gives the kid one of the copies of each of these homologous pairs. And it's a totally reasonable question to ask how it is that the parent decides which of the two in each pair to give to the kid. It turns out, actually, they don't decide. It's totally random. So if we're looking at the father's first pair, there's a 50-50 chance that he'll give the kid this green chromosome or this yellow chromosome with the red stripes. Now, because it is a 50-50 chance, that's basically a coin toss. I actually flipped a coin to decide which of these to give from each parent, and this is what I came up with. So the first one, I tossed heads, and so I said that I would use the one on the left if I tossed heads. So the first chromosome this kid is going to get is this green one from the dad. For the mom, I tossed tails, so the kid gets the salmon one on the right. Similarly, going down the line and using my coin, I decided to randomly put these in. Let's step back for a second and take a look at what I did. So from this pair, the father gave the green copy, here. From this pair, the mother happened to give the salmon-colored one. On the second row, the father gave this purple with a red dot there, and the mother gave the green with the red slash, there. For the third chromosome, the father gave this pink with a red stripe, and the mother gave this yellow one. The important thing to notice here is that there's no relationship between whether the mother gives the left or the right for the first copy, the left or the right for the second copy, and the left or the right for the third copy, and so on down the line. So you could end up with the left from the first line, the right from the second line, the right from the third line, or any other combination. You just get one of each. It's totally random. I've cheated a little bit here and gone and drawn out the rest of what these parents may have given, but you probably get the sense that there are a lot of different ways that this could have been drawn. Okay, so how is it that the mother and father are able to randomize which of the members of the pairs they end up giving to their offspring? The answer to that question is meiosis. Now, meiosis is a special type of cell division, and the cells that meiosis produces are called germ cells. These are more commonly known as egg and sperm. So let's take a step back and just count a couple of things. The father started off with 46 chromosomes. The mother started off with 46 chromosomes, and the baby also ends up with 46 chromosomes. And the chromosome pairs are numbered 1 through 22, and then there are these last ones. You notice on the father that these two don't really look the same. These are the sex chromosomes, so the father is carrying X and Y. The mother has the same 1 through 22, but her sex chromosomes both look the same. They're both these big things. She's got 2 X's. And if you notice here, the baby has 1 through 22 too, and because the baby has 2 X's, that means she's probably going to be a girl. And I say probably because it doesn't always work out that way, but it usually does. And just a reminder, these are pairs. So, times 2. So, 1 through 22 times 2, then X and Y is 46. Let's just take a look at what happens to these 2 pairs of chromosomes here as the father goes through meiosis. And just as a reminder, this is all taking place inside a cell that exists inside the father's testes. Okay, so if we're just looking at these 4, we start out with this genetic material replicating itself.
So I'm just going to keep account of the number of copies of these first 2 chromosomes as we go through these divisions.
So the next step is the cell that these chromosomes are sitting inside of, these duplicated chromosomes, this cell is going to split into 2 cells. Oh, put that 8 down there. So notice, this is 1, this is 2, this is 3, 4, 5, 6, 7, 8. So this cell is going to give off 2 daughter cells, like that. And each of the daughter cells is going to have 4 of these chromatids. Now, chromatid is 1/2 of the X. One important thing to notice here is that in this replication step, there is no new information. So no new info is introduced by replicating the chromosome from this step to this step. Remember when we were talking about which genetic information the baby would get, we talked about the father sending 1 copy of each pair, but which pairs were totally random. So let's see how that works out down here. So this genetic information is going to split up, and it's going to go into these cells here. But whether we have these 2 chromosomes going into one cell and these chromosomes going into the other, or if these 2 chromosomes go into one cell and these 2 chromosomes go into the other, is totally random. Notice that these 2 little ones are not going to go into the same cell. So let's say that the green and the orange end up in one cell up here, and the purple with the red polka dot and the yellow with the red stripe end up in the other cell down here. You'll notice that I keep drawing these not really as X's, even though they come out looking like X's, but I draw one arm and then the other arm. The reason that I'm doing that is that this thing that I've drawn here, for instance, is actually a complete chromosome, and this thing is another complete chromosome. They're copies of each other, but they're just bound together in the middle.
Chromatids.
So notice that if I draw the same line here as I've got up here, and we could start thinking about the difference between genetic information and the volume of genetic material, each one of these cells has the same amount of genetic material for chromatids as this cell does, but they actually have half as much information because they just have 2 copies of the same information up here. This is like if you made a photocopy of a document and stapled the 2 things together. You might have 2 sheets of information, but you got the same information on each one. These cells are going to go through another round of division. So in going through this division, these cells are going to cut the amount of genetic material that they're carrying in half, but they don't really cut the amount of genetic information that they have. And the reason why that's the case is that these chromatids separate, and then because they're copies, the same genes end up in each of the daughter cells. So this one gets one of these long green ones, and this one gets one of these long green ones. Same for orange, yellow with the stripe, and purple with a dot. So notice each one of these daughter cells has a copy of one of the 2 long chromosomes that were in the first position, and one copy of one of the 2 shorter chromosomes that were in the second position. So here it looks like we've got 2 different kinds of cells. We've got long green and a short orange, and then this one with the yellow and the stripe and the purple and the polka dot. And remember, because I made a random choice here, this could have ended up differently. This orange could have ended up with a yellow with a stripe, and the green could have ended up with a purple and the polka dot. So we could have had 4 different possibilities. But now we were only looking at 2 of the different types of chromosomes. So we're just looking at 2 pairs of 2 of the chromosomes. If you were to take into account all of these chromosomes, there are 23 pairs, remember, you'd be able to get 8.5 million sperm that had unique combinations of these chromosomes. I mean, when you think about it, at this point, we're not doing a whole lot of biochemistry. We're just saying that these things can combine randomly, and they can combine randomly in a whole lot of different ways. I think it's pretty impressive that we can get so much variation just by randomly recombining these things. Now, you remember that I started off saying that this was the father, and that wasn't just because I was specifically using the father's genetic material. This system looks a little bit different for the mother. So this was the father. Now let's take a look at how this looks different for the mother. So to show you how different things are, I'm just going to go through, I'm going to start out with this copy and paste of what happens with the father, and I'm going to erase the things that are different for the mother. So the mother doesn't end up with as many cells coming out here. And here, when I drew them for the father, I drew them looking pretty much the same because they're really similar. But that's not what happens with the mother. So up to this point, the father and the mother do look pretty similar. So when the mother's cells are undergoing meiosis and producing germ cells, one of the germ cells comes out pretty big. The other one, though, comes out really small. This cell here doesn't have enough machinery to be able to survive. It carries all the genetic information that the similar cell with a father had, but it doesn't have enough endoplasmic reticulum or mitochondria for this cell to be able to divide. So even though these both have the same amount of genetic information, this cell, this thing that we call a polar body, it doesn't survive. It dies almost all the time. So this big cell here that's taken up most of the cellular machinery so that it can survive and produce all the things that it needs to live and develop, it splits one more time. But again, it splits into really asymmetric cells with one big one and one little one. So this one would have 2 of the chromosomes if we're just following 2 of the mom's chromosomes. This one would have 2, but again, this polar body down here doesn't make it.
It dies off.
So let's remind ourselves of why we're doing all of this. It's really important to be able to get half of this genetic material into the next generation. There are some people who end up with some extra genetic information, and especially common place that happens is here on chromosome 21. So you can have a person who, instead of having 46 chromosomes, has 47 because they've got this extra copy of chromosome 23. This is a condition called
Trisomy 21?
and it's more commonly known as Down syndrome. The reason that we need to go through meiosis like this and avoid conditions like Down syndrome, which are very dangerous, by the way, being able to control the amount of genetic information that goes into the next generation is a crucial step in propagation of really any species, not just humans. Most animals have some sort of system like this to control the amount of genetic information that they're passing to the next generation. There is one last point that I want to make in going back and looking at this part here. Now you might look at these 2 cells, the information that I've put up for these 2 cells, and think that they're identical. It turns out actually that they're not, because there's something that happens here where the homologous chromosomes will actually swap DNA with each other. So in this process of recombination, we might want to consider these 2 big chromosomes here. When they're in this big cell with the 8 chromatids we were looking at, they're actually touching each other. They're actually wrapped around each other. So it might look a little something like this. So what happens is the backbone on the DNA will actually break on both of the chromosomes. Remember, each of these chromatids, has one long strand of DNA running through it wrapped around some proteins. So if you break this thing, if you just chop it on both of these chromosomes, on the yellow one and on the green one, all of the DNA above here, all the alleles would switch over to the other chromosome. So what this comes out looking like is this once you pull these things apart. These 2 chromosomes have actually swapped part of their arms, but they only exchanged information in one place. Because these are such long molecules, they could have been crossing in multiple places. Not only that, these arms on this side and on this side also could have been in contact with each other, and they could have exchanged information. What I'm describing here is actually more properly called homologous recombination because the chromosome pairs are so similar to each other. Even though you're breaking the DNA, both of these chromatids come out with the same amount of genetic information because they both break in a similar place along the length of the strand of DNA. So there's also no loss of information to either of these chromosomes. They come out with all the same genes. It might be different copies of the same genes, different alleles of the same genes, but they all end up containing the same type of information.